Kia ora mai koutou. Ko Hanareia Ehau-Taumanu no Greenlaw. He iri ahau no ngātu e pohatu, ngātu pura o tōna pune me tātou hoki. Ka koutou ko ngā mihi I ko mihia. I runga I te kaupapa nei. He kai mahi ahau the rangau ahi marakai, no bioprotection Aotearoa. And I love microbes. So you're going to keep hearing about them, even though we've had 2 fantastic talks about that. Please don't expect as much from mine. These wonderful PhD students do so much work in their studies, and I finished my grants on Sunday. So this is very, very preliminary mahi that I've been doing. It's taken a lot of bioinformatic processing for me to get to this point, and so it's a very preliminary snapshot, but I will talk about what I hope to do in the future. And I also just want to put up the hierarchy that I used the lovely kupu that have come out of the mahi at Sion, because I've always wanted to find a kupu Māori from the front, and now I can. So kia ora. So I've been working on Myrtle Tawake, which we got a huge introduction yesterday morning about it. When I returned home to Aotearoa, I had no idea about this tree. It was first introduced to me by my supervisors and like, you're going to do a bit of rust. Have you thought about Myrtle Tawake? And I was like, yeah, what is it? So I went out into the ngahere, into the swamps and found it. And I have become a huge fan and a lover of this tree. I like to show people how cool it is, particularly the pneumatophores, the cool little roots that come out of the water. Kids love it. They're like, oh, it looks like an alien. And I'm like, no, it's just helping the tree breathe. So it's an amazing organism. So yes, I've been working on Myrtle Tawake. As we all know, it's hugely susceptible. It has beautiful flowers and the fruits are a little bit tangy. So Myrtle Tawake, as introduced yesterday, is throughout the North Island and the top of the South. Another wonderful PhD student who's just defended was looking at the genetics of Myrtle Tawake across the North Island. And so in this graph here, you can see on your right, the most northern populations that they sampled. So they're kind of clustered together, so they have similar genetics. And then the ones on the right, on the left, are more southern, so that's the Mano to Wairarapa area. And so for me, as I love microbes, I wanted to know, just like Vlad and those before, what other roles that these microbes could be playing in terms of disease protection, and could this also be a risk? And so this project was, it's just really an exploration, continuing on a lot of mahi of others, to really get into not just the taxonomics, so who's there, but I want to know what they are doing at the same time. So what did I do? I was able to take some cool pictures with the document. So I went out into Ngāhere in Tāmaki, Taranaki, and the Wairarapa. So Tāmaki is Auckland. Taranaki is the beautiful Maunga up the west of the North Island, and then the Wairarapa is close to Wellington. I didn't put a nice map like a grown-up's had, but that's where I went. So very different geographical regions. And I went out and tried to find Myrtle Tawake trees that were both asymptomatic. So I can't say or not if they had myrtle rust at the time, but then I also found symptomatic leaves where you get the nice pustules and collected them. So I collected them from the bottom of the canopy. If you know Myrtle Tawake, it is not a short tree if they are old. So I had about 8 meters of reach, or if I stood on a branch which broke health and safety, I could get to maybe 9. So I spent a lot of time with my poles going around the ngahere trying to collect these leaves, and I did this over the 2024 summer. So it was just when myrtle rust was present. So it was actually quite hard to find asymptomatic trees, but as you've heard, they are there, and it just takes you a little while to treat them, as you all know. So what did I do after that in my Airbnbs? So I get the samples from the field, I'll take it back, and then I would put the leaves in bags and buffer and sonicate them. And so this is to get all the microbes off the leaf. It's like high sonication, shaky, and all the microbes that fall on. Because that's what I want to know. I want the ones on the surface. I haven't studied endocytes lab, but I will get there one day. But this is just getting the ones off the leaf, because that's where myrtle rust goes first, right? It's on the leaf surface. So I did high sonication, and then I did a lot of filtering of that buffer. This is my lab setup there, which was cool. And then, collected the DNA from the microbes that were in that buffer, and then did sequencing. The sequencing that I did is a bit different from the last couple of presentations, which was looking at a particular gene within the microbes. My sequencing was doing the whole genomes, or all the genes that could be sequenced through these microbial communities. And then I did a bunch of bioinformatics to look at taxonomic classification and relative abundance, because this is as far as I've got, but it's enough in itself. So getting right to it, here are just the Tamaki, Makoto, and Taranaki samples. I got about 5 Myrtle from each of those, and of course asymptomatic and symptomatic. So just here is the asymptomatic one. So this is what we might assume is healthy, but they just showed pustules or spores on their leaves when I was collecting them. And as you can see, this is just the relative abundance of the taxa that have been identified, and they have a lot of complexity across them. Most of these are bacterial. It is hard to find fungal reads in a lot of my samples. They are there, I'll show them soon. But there is quite a diverse set of bacterial genera that have been found. As you can see down the bottom, there are those major ones there. So that's your Pseudomonas. That is a very common leaf inhabitant throughout the environment. And then also Streptomyces and a few others like that. So as I said, I'm investigating this just as you're looking at it at the same time as me. But it's nice to see that there are some trends, but there are also differences. And these are for individual trees. But you can see even though you're in Taumaki in the north and you're in Taranaki in the west, there are still some similarities that those trees are holding on to. So this means that the Māori Tawaka is likely selecting these types of microbes for their benefit, and that they actually play an important role in the functioning of these trees as well. However, when we go to the symptomatic ones, you assume that toxicity would be everywhere. And it didn't see as much. These are still just bacteria, so you can't really see it, but the whole thing changed. Instead of seeing lots of bars where you can see, yes, there's lots of microbes present, it suddenly went to only a few. So therefore, Myrtle rust is likely having an impact during its invasion on the leaf. And these are the only ones that are being able to survive the competition that might be going on in those leaf surfaces. One of my favorite microbes, Pseudomonas, was still gradually there, and it's known to be quite antagonistic to other bacteria. I don't know if it's a good antagonist of Myrtle rust, but we can find out. But then also Patoa just really took over, and that is also, which Lad also showed, can also be an antagonist as well. So it seems like the bacterial genera that are highly abundant could be playing a role in terms of trying to antagonize Myrtle rust. And then the others that we were seeing in the previous slide are either just really at low abundances or just not found in what I was able to sequence, but might not be, but be more impacted by Myrtle rust over time. So again, it's just showing the differences between them. And obviously, there's a few differences over what is there, but overall, really showing just that huge decrease in the taxa that I was able to find on these leaves. So there is an impact. And so just to show you those two slides together so they can get an idea of all the asymptomatic versus the symptomatic in both regions. When you can see the impact of infection to those that were asymptomatic. And again, so you just see the huge complexity that was there. There's still a lot of reads in the grade, or they're just very low. That could be other taxa, but it's just hard to find it in this type of sequencing. But you can see that I'm still getting the majority in those other samples that actually are infected with Myrtle rust. So this was really interesting to see. I knew that diversity would probably decrease because that's what pathogens are great at. They compete, they get in, they take over, and others just can't survive. But I didn't think it would get to this impact within the type of sequencing. Oh, this is not my slide. Hey, come on. Oh, my God. No, okay. I'm just going to describe to you what would have been on my slide. So I went into the fungi, and so if you took all the sequences that I had, and say I found about 2/3 were bacteria, and then I got probably like 1/16 were fungi. And we know that the fungi are important because Myrtle rust is a Basidiomycota, so we were looking at those to see if they're antagonists. So I was going to show you these plots that were showing that 1/16, so that you could see that Ascomycota and Basidiomycota, which are two major fungal phyla, were present. And then there was actually a lot more diversity for the asymptomatic leaves. So you could, again, similar to the bacteria, you could see multiple taxa were present. Some of them were known antagonists that had been found in Brazil, so that's also really interesting. We either have similar taxa that are present there just because we know microbes go around the world, or they could potentially be something that is endemic to Aotearoa that could help tie Myrtle rust as well. So there's lots of diversity there, and I was going to show you the pretty pictures and diagrams, but then it really changed when you looked at the symptomatic samples. So again, you can see lots of Vaccinia. So that was the genus that Vaccinia used to be part of. So I think the databases need to be updated. So the genomes will definitely help. But yeah, you could see it really shifted. It was actually picking up other Vaccinia as well. So I'd have to do a bit more of a deeper dive into what is actually there. But one of my trees, it literally in the asymptomatic had all this diversity. So imagine a circle and it had lots of things, and then suddenly half of that circle was just Vaccinia. So again, it's just showing that impact that Myrtle rust is actually having just on the leaf surface. Remember, this is just the surface of the leaves where the Myrtle rust is actually present. And I must say, the samples that I took, they weren't fully covered in the rust pustules. You could still see green parts, so it's not like it was like a dead leaf that had curled and it had everything on it, which I would assume would only be Myrtle rust coming through. These were more of a kind of a 50-50 in terms of the coverage on leaves that I was sampling, because I still wanted to see the other microbes that were present. So I'm very sorry that you can't see my slides. They were very pretty. That's what I did on Sunday. So yeah, so it was great to see that there's this impact, right? It's a very preliminary snapshot. I'm going to do much work, so hopefully I can present it to you at another time. I'm going to look and see my money. But this is actually really part of a bigger project that I'm working on. And so what I plan to do next, again, is to continue to go through the data and really seeing if there are any antagonists present. So kind of like Dan, who was looking for the hyperparasites, really seeing if there's anything else out there. Scleropsis has been shown to actually be an antagonist. And in Australia, there was an endemic species just recently found and identified. So maybe we could also have something similar here in Aotearoa. So I'm going to do a lot of money in that. But I've also looked at the leaf microbiomes of other Myrtaceae species. So that would be Porticoa, Manukata, Metrosis perforatus, that's one of our other vines, and also Fijoa. We all love our Fijoa, and it's part of the Myrtaceae whānau as well. So I've done all of that as well, because I want to see, just like Vlad, is the plant microbiome also playing a role in the resistance that we're seeing in those more resistant plants? And can that provide protection to our other Myrtaceae plants? And so I've actually done those microbiomes as well, and we'll be looking at that susceptibility kind of range, but through a microbiome lens as well. And then Black has some great questions at the end of his presentation, which I also have. And so recently, which I don't have the data for because I only just finished it before going overseas, but I actually took the leaf microbiome that I collected from the species in the middle and applied them to Myrtle Ehau-Taumanu, either as a preventative or a curative treatment with the infection in between. So I had these beautiful Myrtle Ehau-Taumanu seedlings from Tangitani or Manawatu to actually test that. So I went into the greenhouse and I applied these microbiomes, I sprayed them with the rust or vice versa to see And it was also similar to what we saw this morning, where the preventative effect was actually greater than the curative effect. So it's really, for me, that indicates that the microbiome can play a role in the protection against myrtle rust. And it might be actually diving down more, which I hope to do, to actually finding out who are those key players, what function are they playing? Are they antagonizing myrtle rust? Are they supporting other microbes that can antagonize myrtle rust? And all those kind of connections that can actually happen in the microbiome. So yeah, there's a lot more work and a lot more mahi to complete this kind of story for Maere Tawake. But as you can see, it's really in the hopes for all of our rotating species down the line. This is not my slide either. Aroha mai. But in summary, you can see that there was that huge difference between the asymptomatic and symptomatic microbiomes of the leaves of Maere Tawake, and really showed that myrtle rust does come in and really takes over those communities. I am going to do more analysis in the fungal players, and as you can see, I have a lot more to do in this mahi that I am really going to try hard to make sure I can do to the full extent of my abilities. And so for this mahi, I just want to acknowledge Ngā Samoho e Ngā Ruahine, where I did these samples and are in communication with to give back my results. And then also to the farms that actually allowed me to get access to these trees, because usually you have to go through someone's backyard to get the banana in the first place. And with that, thank you for listening. Kia ora.